goes, and like if a stray dog comes up to him, all he has to do is pull the trigger. Like he doesn't even have to, and the dog turns around, goes the other way. What's that? Yeah, he likes it. He's like, this is great. Stop thinking how to shoot him. Mike, thanks, Dad. All right, we're going to go ahead and get started. It's a little bit early, but we got a lot to do today. So we are going to finish up viruses, and then we are going to have an exam review, and then we're going to jump into epidemiology. Chances are today we're not going to get to the epidemiology activity. The activity will probably come on Tuesday of next week, just because we're going to run out of time. Second, I am aware that Heather misunderstood the instructions for entering grades in for the practical, and you guys have like F's for some of you. Whatever your grade is, all it needs to be is double. So if she entered it out of 50 points, literally she just needs to multiply whatever grade is in there by 2, and that will be your grade earned. So she can update that for you. I did add, for those of you who came to clean, I added all your points on top of that. So when she doubles that point value, that will be accurately reflected. From what I can see, she only did that for one class. The other class has accurate grades. I was like, what happened? So Heather will be updating those. We have the lab exam for those of you who are in the evening section tonight. Don't forget, please come to class. Anything else? Exam review for next week. Oh, the exam next week is on Thursday, right? Everybody knows that? Yes, exam is on Thursday. Ooh. Next week, you will be starting your microbe isolation project in lab. So that this is done consistently, if you have a laptop or a tablet, it is recommended that you bring it to lab on Tuesday. If you do not have a laptop or tablet, we will have loaner laptops for you to use. But they're the colleges and they're not the greatest, so they're kind of slow. In the microbe isolation project file, you will see a document, a Word document. That Word document, the research portion must be filled out before you leave lab on Tuesday. So that's what you're going to be using lab time on Tuesday doing, researching and filling out the research portion of that document. And then other than that, the process will be fine. All right, so we left off virology talking about characteristics of viruses. So somebody give me a characteristic of a virus. What is one of them? Envelope. Yeah, can be naked. Envelope, non-envelope. What's another characteristic? Non-living. Excellent. DNA or RNA. Nucleic acid. What else? Relies on the host. Obligate, intracellular, parasite. Any others?
Doesn't respond to stimuli.
That goes with no life. What else do we got? Everybody probably forgot the helical capsule versus the icosahedral capsule. The thing that looks like a dye versus the thing that looks like little crocs kind of lining it. Remember, replication speed is directly influenced by the size of the virus. So, if you have a very small virus, the replication speed will be very fast. If you have a very large virus, the replication speed will be very slow. So a DNA virus, a large DNA virus, will have a longer asymptomatic time frame than a short RNA virus, because it has to make all those viral proteins. So why am I talking about this? Well, today you are going to learn positive sense and negative sense. So these are two different classifications of RNA viruses. They're very important to understand. The single-stranded RNA positive sense is the exact same charge as your mRNA. So the mRNA your body makes to make proteins has a positive charge. So if a virus has a positive single-stranded mRNA or RNA virus, it can immediately sit down on our ribosomes and make viral proteins. Immediately. So as soon as the virus enters the cell, it becomes infectious. Versus an antisense virus. This is the opposite charge. So what ends up happening is it has to have a viral polymerase that codes or makes a positive strand copy. So this is one additional step that the virus must do before viral proteins can be made. And it must keep making those copies for it to be effective. So one of the targets when you're creating antiviral drugs, which we'll talk more about in chapter 20, is to target the protein or enzyme that turns a negative sense into a positive sense, inhibiting its transcriptase. Because if that does not get turned into a positive sense, it will not make viral proteins, you will not make viral particles. So, positive sense, take home. Replicates faster. Because it doesn't have an additional step. Alright, so this is a breakdown of the life cycle. So with a positive sense, so blue should be, let me see, I can't really make it out here. Yeah, blue is your positive sense cycle. So the virus goes in, or I'm sorry, black actually. Positive sense. Why are we showing all three pathways? Okay, this is double-stranded RNA. Ignore yellow for right now. So if we're following the black arrow, which is the innermost arrow, we have the virus attaching, going in, and immediately creating RNA proteins. Now it does that with the ribosomes that are naturally free-floating in the cytoplasm. The RNA negative sense or antisense has an extra step. So the viral genome has to first make a copy of itself in the positive sense. Then the positive sense sits down on ribosomes and starts making viral proteins. So what kind of viral proteins are we making? This is where it gets complicated. You have two terms when you're talking about viral proteins. Structural and non-structural proteins. A structural proteins, super easy. Attachment proteins or glycoproteins is what you'll read about them, or capsid proteins. So how are you making that icosahedral or helical capsule? With structural viral proteins. How are you making that attachment or glycoprotein? Structural viral proteins. So what are examples of non-structural viral proteins? Well, we talked about herpes last time, and how herpes makes a viral protein 16, VP16. The sole purpose of VP16 is to make sure that nerve cells bring out their DNA replication machinery so it can make copies of itself. That is not structural, but is required for the virus to replicate. Okay? The enzyme that takes a negative sense virus and codes a positive sense copy would be a non-structural protein, because it's adding to the replication of that virus. Okay? So, when you are writing your genetic nuance paper and you are doing it on a virus, you need to pick either a structural protein or a non-structural protein name the gene that makes it, and was its function. It's just that easy. Okay. This is going into more detail. Basically, attachment goes in the host cell. This is positive sense, and it makes viral proteins. This is the negative sense. You can see that before it starts making viral proteins, it must transcribe a positive sense copy. And then this is double-stranded RNA. All right. We talked a little bit about hepatitis C, and then the way you transmit hepatitis C is through IV drug use. I'm going to give you a statistic, and I want you to generate some ideas. Some of you will probably get it right, but most of you are going to get it wrong, so don't be afraid to be wrong. Because a lot of people argue about this to this day. The sex ratio of hepatitis C, 75% male, 25% female with chronic hepatitis C. Why do you think that is? Remember, number one way you get hepatitis C, IV drug use.
They get more IV drug use.
Males generally have higher risk-taking behavior, which generally means higher rates of transmission of Hep C. That is true. There's something else going on.
They're having sex with each other.
That is also true. And that sex would include blood. So, but that's not what I'm going for.
Repeat the question again.
Don't get treatment. So prior to 2015, there was no effective treatment for hepatitis C. Now we have a cure. The problem with that cure is it's $100,000. So I don't know about you, but your average heroin user is not gainfully employed with health insurance, or has so much money that they can pay for the cure.
They're infecting other people. They know they have Hep C and they're sharing needles without telling people. So that's how they're higher. Do women clear it easier?
Yes.
Yeah.
Why do women clear viruses specifically faster?
Is it because we menstruate?
I mean, kinda. It's because you gestate life.
Yeah.
You have to. If you don't clear the viruses faster, we don't have a next generation. So, what is the science behind this? It's not just the magic of menstruation cures all viruses. It's not like I'm taking menstrual blood and like, I'm going to cure hepatitis C and come out like formalin. So, what's going on? So, there is a cell receptor called a toll, T-O-L-L, hyphen, like, L-I-K-E, receptor. So when you think of a toll, what do you think of? Right, you have to pay the troll toll, right? You gotta go over the bridge. You're the only person that got that joke. So you gotta pay the toll when you go over the bridge. A toll-like receptor, therefore, screens things within a cell. So women have a faster toll-like receptor that screens for double-stranded RNA. So hepatitis C is a negative sense virus. That means when it makes its copy of itself, its complementary positive sense, it creates what? Double-stranded RNA for a minute. And the second that happens, the female body is like, you're out of here. Completely disassembles it. And that's because if they didn't, the next generation would be harder and harder the fertility rate would drop. So there are a few things like that where the female body has to respond faster or it will directly impact the infant. So if you see differences in sex in diseases, first question you should always ask, what's the behavior that gets you there, right? So like when we're talking about specifically IV drug use, we know that males take higher risks on average, and we know that there are a larger proportion of IV drug use. So therefore, of course, you would see higher rates of hepatitis C within that population. That is the accepted kind of talking point. But when we see women, so we take a cohort of women, IV drug users, we take a cohort of men. Say we take 100 women and we take 100 men. They're all hepatitis C negative at the start of this study. We test them every week. And we notice that the same rate of infection occurs between the sexes. So you're already taking risky people. So they're in it, and they're riding the H train all the way. Then you start to notice that as they all get hepatitis C, females clear it at a higher rate than males, and then that's where we get to the cell receptors being uniquely physiologically different.
So would that mean the men would have a more severe withdrawal?
Don't confuse the drug with the disease.
Okay.
So no.
Okay.
Like,
Two different things.
there's two different things. We're specifically looking at a female's ability to identify double-stranded RNA
Okay.
and act on it faster.
Okay.
So it's kind of a fun little fact, and it kind of takes home why these steps matter. So you know that if you're working with a negative sense virus, you're going to see different disease rates between the sexes. Because just a double-stranded RNA virus, women are intrinsically going to clear it or be asymptomatic more likely. Now men, we have a few advantages too. I'll explain it. It's a little bit of a, it kind of sucks, but we're going to get to a few of the diseases that we can get that we just has no effect. Like most of the STDs will yield a female, if gone untreated for a long enough period of time, completely sterile. So if you have an untreated gonorrhea and chlamydia and you go for years, you can have pelvic inflammatory disease if you're a female, and that can lead to sterility because of scarring. No STD on the planet can make a man sterile. They just continue acting like an STD dispenser. Chlamydia for you. Everybody check under your chairs. It's chlamydia! All right. Picorna viridae. So one of the other ways that we classify viruses is by size. So pica meaning small. So enterovirus, poliovirus, rhinovirus, and hepatitis virus. All of these are naked, positive strand, non-enveloped viruses. When I say naked, your first thought should be that alcohol-based hand sanitizers will not be effective because there's no envelope for it to degrade. So, everybody's familiar with rhino, common cold, right? Hepatitis A, we talked about that last time. It's acute inflammation of the liver. And Coxsackie. Coxsackie virus is hand, foot, and mouth. So if you have kids that have ever had hand, foot, and mouth, this is painful blisters on the palms of the hands, soles of the feet, and in the mouth. Very painful lesions. So daycares get this that have carpet and stuffed animals that are fur. Because one kid will bring it into the environment and there's no amount of cleaner that you can use safely on the carpet or those stuffed animals that will remove that virus from the environment. So like St. Agnes had an outbreak in their child care center in 2016, and they called me in for a consult. They said this is the third outbreak of hand, foot, and mouth that we've had in our facility in the last 6 months. And they said, come in and take a look. I was like, rip out all of this carpet, get rid of those types of toys, and this is the cleaner you use. I'm like, cool. So if you ever go to St. Agnes and we're like, why are the kids playing on linoleum or rubber? Because it can be sanitized and disinfected. So if you're going to like a large daycare and you see a lot of carpet, they are dumb. They are going to have lots of outbreaks of Coxsackie virus as well as norovirus, which is in a different family. But everybody in this family member, they're small viruses. Pico, Picornaviridae, positive strand, and single-stranded, non-envelope. Next one, Togaviridae. Togaviridae is named after it because of the size of its envelope. Togaviridae means the envelope is quite big. So what do you think the impact on viral survival in a large envelope is? No ideas? More dangerous for what?
For them.
Yeah. So if you are wearing a large Halloween costume and you are trying to walk around and do daily functions of life, you're going to get caught on things, you're going to get rips, you're going to get tears, you're going to be inconvenienced. As a virus's envelope gets bigger, the likelihood of it inactivating increases. So if we look at these viruses, specifically the alpha viruses, this is like chikungunya, which is spread by mosquitoes. The only way to get that virus is if the mosquito directly injects it into your body, because the envelope is so fragile that if it dries out or gets exposed to oxygen, it will literally just fall apart. So Togaviridae. Rubella is very similar, but it's not transmitted by insects. Rubella is airborne. Rhabdoviridae. Now, Rhabdoviridae, this is rabies, is negative sense. It's a Lassa virus. We are very happy that this is a negative sense virus. Because if it was a positive sense virus, humanity wouldn't have survived much longer. Do you know how many days you have to get treatment for rabies before the treatment is no longer effective? 48 hours. You have 2 days. There have been 3 females. What did we say about females? Yes. But which type of infections? Yeah. The only 3 surviving cases of rabies without medical intervention were all teenage girls. Because, ladies, why? You clear the double-stranded RNA faster than men. 0 male survivors ever of untreated rabies. 0. In the history, since we've been keeping surveillance, we've had 3 female survivors. Now, calling them survivors implies that they were fine and nothing is wrong with them. They are not fine, and they have serious brain damage as a result of being infected with the virus. So they are alive. One of them has just very low speech. She can think quickly, but her speech is very slow. Another one has limb paralysis as well as speech issues. But anyway, they didn't get off scot-free. It's not like, ladies, you are now immune to all rabies. Go out and play with and frolic with bats or whatever. Don't do that. Don't poke the possum. Yeah, possum bites. Yeah, so that's rabies.
So it affects your nervous system?
It does. So it replicates first in what, so the closer you are, the closer to the brain where the bite happens, the faster the virus So the best option is leg or hand. That buys you the most time. If you get mauled in the face, and the virus has literally got nerve tissue right there that it can grapple onto. So it specifically has tissue tropism for the central nervous system, and it looks like a bullet, and it will go straight through your brain and you're dead.
Isn't the vaccine for it or the treatment for it based on weight?
It is, and it can kill a diabetic. The prevention. So the treatment for this is weird. First, we give you 2 rabies vaccines. And you're like, bro, doesn't it take like 2 weeks for a vaccine to work? And I'm like, yes it does. So we give you 2 rabies vaccines, and then at the site of the bite, we inject you with immunoglobulin antibodies, human antibodies to rabies. You're probably thinking, where do we get these human antibodies? They harvest them from veterinarians, animal control workers, and other people that have to get the rabies vaccine as a part of their occupation.
So
So-
they're still alive that they harvested from, right?
Yeah, there's donors. It's like a donor list.
Okay.
Like when you get the rabies vaccine, you have to sign up to be a donor. And they will, and that's what they administer. So they give you antibodies from somebody else that immediately start working. Then they give you a vaccine for rabies, which will take a while to work. So they have an instant, but they can't give you the vaccine and the antibodies at the same location. Why?
They don't fight each other.
The antibodies will neutralize the vaccine. So you have to and it's about $1,700 for each trip, and you have to get your shots day 1, 7, and 20. I can't remember the time sequencing, but you're doing this 3 times. And it causes such an electrolyte imbalance and shock that diabetics have a high negative outcome as a result of that treatment. So, you're diabetic, your high-risk animals are foxes, skunks, bats, raccoons, possums. Just don't. Just don't. All right. This is what rabies looks like. It's got a helical capsid. It looks kind of like a bullet. And that's what it does. It just rips through your central nervous system. Ritoviridae. Now this is where things get weird. This is called an orphan virus. Does anybody know, and I don't expect you to, does anybody know what happens when a virus is orphaned?
It has nothing to do with it.
Kinda. So, when you find a virus in a tissue, but the virus doesn't appear to cause any damage to the tissue, we refer to it as an orphan. Because it appears to be replicating but not harming the host in any way. What do viruses do by their very nature? Cause a cytopathic effect or damage the host. When they don't, we call them orphans. So this is a respiratory enteric orphan, which means you can isolate it in sputum, you can isolate it in the gut, and it's not causing any damage, it's just chilling. We don't know what it does. Rotavirus. This is a weird vaccine. All right, we're going to introduce a concept that I need you to start to understand as we talk about these viruses, and that is the concept of seroprevalence. Remember, your serum contains your antibodies. Your antibodies is your unerasable web history of every disease you've ever come into contact with, and ever will come into contact with. So we refer to it as seric conversion or conversion when you are exposed to an organism, either through vaccine or in the environment, and then you create higher levels of those antibodies where you will test positive. Rotavirus can cause severe gastroenteritis in young children. So much so that it will cause hospitalization and death. This virus has a double capsid, so it is really hard to remove from surfaces and disinfect. It's got 2 shells. Now, there are parents out there that are against childhood vaccines, all of them. And so regardless of whether or not you get your child vaccinated for rota, they will get rota. Okay? What I mean by that is we evaluated a group of kids that were unvaccinated with rotavirus. We took blood samples and we followed them for several years. 100% of them had antibodies to rota after the age of 2. So the rotavirus vaccine is designed to keep your kid out of the emergency department. Because that's what will happen. The kid will dehydrate and you will have to go to the emergency department for IV fluids if you have one accessible. If you don't, your kid will die. Now, when you think of most vaccines, we've started talking about a few myths. Chickenpox is not a vaccine. It's a controlled infection that colonizes you with a virus for the rest of your life. Rotavirus vaccine is not a traditional vaccine. It's only designed to get you to the age where you can survive the rotavirus on your own. So we give it to you as an infant, and we give you a few boosters, and then when you're 7 or 8, a little diarrhea is fine. Here's your Pedialyte, here's your Gatorade, you're good to go. All of you get rota every 2 years. You're like, why did I have that weird loose stool? Oh, you got reinfected with rota. It's everywhere, constantly in the environment, and because it has a double capsid, no amount of UV light will destroy it. It's just everywhere. Literally on everything. So when you're talking to a vaccine-hesitant parent, you can say this. The child will have immunity after the age of 2, regardless of whether or not you get the vaccine. You are taking the risk of the child dehydrating. That's really it. So often first-time parents that think they're educated will go, I've done my research, and then they will probably experience a dehydration event. Then the next kid will probably get the rota vaccine, because that is a scary, scary situation. Question? Yeah.
What's it called, the vaccine?
It's Rotavax. It's something stupid. It's in the name. You know how people name it? It's like Rotarix or Roto-Rooter or something. It's Rotrex. No, it's Rotrex. Like yeah, Rotrex. Like it's yeah, Roto-Rooter, so you don't get Roto-Rooter. So not all vaccines give you the immunity that you think they're giving you, because you were taught at a very young age, vaccinated immune. Sometimes vaccinated just so you don't die when you get it. We only need to get you to about 13, and then your body can pretty much fight off most things. That's why the number of vaccines that we give you drops off exponentially as you get into your teenage years. Because you can survive it. You're good. But we're just trying to stop the babies from dying. All right. Single-stranded RNA produces DNA. These are reverse transcriptase. So reverse transcriptase viruses, retroviruses, they start out as RNA, they make a DNA copy, and sometimes that DNA integrates into the host genome. Hepatitis B is like that, HIV is like that. Those are the two most common reverse transcriptase or retroviruses that we have in the human population. Now, if you're thinking critically about this, we have a vaccine for hepatitis B. Why don't we have a vaccine for HIV? Okay, let's talk about what reverse transcriptase viruses do. RNA makes a DNA copy. That DNA copy then gets integrated into the host DNA. With me? Every viral protein and every copy of the virus now made is using your DNA replication machinery. So that means low mutation rate, easy to vaccinate for. Okay? That's the logic. HIV has one of the highest mutation rates of any virus on the planet. Somebody troubleshoot for me and tell me which stage in the process is responsible for the mutation. Your gold standard is hepatitis B. Hepatitis B has a reverse transcriptase, integrates into the genome, creates flaws. So where is the problem for HIV?
Is it in the RNA?
No.
Integrating the DNA.
No. No, it wouldn't be when the DNA replicates because they're using our machinery. We make perfect copies. We're amazing.
Is it the copy of the DNA they make?
It is. It's the reverse transcriptase. So, hepatitis B's reverse transcriptase holds on with 4 bonds. HIV's only holds on with 3 and constantly falls off of the viral RNA, creating random mutations. So because it does less, it mutates more, and that makes it more unstable and harder to create a vaccine for. If HIV's reverse transcriptase held on with 4 bonds, HIV would have been eliminated in the 90s. But because of its mutation rate being so high, we have yet to create a stable vaccine for HIV. Our greatest success is a 5% efficacy vaccine.
Do you think they'll ever make an efficient vaccine or?
With any vaccine, the important part is finding a spot that is outward facing on the virus that is stable. If you can't find a stable outward facing component, you will never make an effective vaccine for it.
So because it mutates so much, it's really hard to find that?
Yep.
That sucks.
And it can't be a stable part on the inside of the vaccine because once the vaccine uncoats, you're already infected. So it has to be on the surface. Okay. Oh, I used this term last time, oncoviruses. Oncoviruses literally mean any virus capable of giving you cancer or causing cancer. So the reason that we went over why warts cause cancer, they kill the part of your cell that can engage in apoptosis. HIV can cause cancer because it suppresses your T cell response, which is your cancer response. Hepatitis B can cause cancer because it integrates its genes into your DNA, which can also result in damage to apoptosis, and you get liver cancer. This is just going over the reverse transcriptase pathway. Remember, there are 2 types of ways that a virus can leave a cell. One is they fill up the cell with viral particles and then lyse, and the other is they slowly push out virus via budding. In order for you to be chronically infected with something, it needs to be an enveloped virus that engages in budding. If it doesn't, you can't be chronically infected with it. All right, viruses and cancer. Several types of cancer are caused by viruses. They may develop after long viral infection. Carcinomas, sarcomas, and adenocarcinomas. The number 1 cause of cervical cancer is HPV. If we eliminated all the oncogenic strains of HPV in the world, we would save about 9,000 lives in the US each year. About 2,000 colorectal cancer deaths, about 1,500 penile cancer, and about 1,000 cancers of the head and neck. So if everybody was vaccinated, we eliminated HPV from the planet, at least the ones that cause cancer. In this country alone, we're looking at about 15,000 to 20,000 lives. And these are lives that matter. So these are people between, I mean, you can get the cancer as young as 25. So we're looking 25 to 57. So these are economically productive people that you are losing when these cancers did it. All right, oncogenes transform the cell into cancerous cells. Oncovirus become integrated in the host cell genome, make tumors. A transformed cell harbors something called a tumor-specific transplant antigen, or TSTA, on the surface, and T antigen on the nucleus. So you probably, if you have been paying attention to medical news, found out that we're developing an anti-cancer vaccine. And this vaccine would literally take your T antigen from your type of cancer, and it would make a custom vaccine for you. And then your body would cure itself of its own cancer. This is not the type of vaccine you take prior to getting cancer. They literally make the vaccine for your specific type of cancer using that tumor antigen.
Is that when they think they're chemotherapy and whatnot?
So we don't know yet. This is not proven. Yeah. So we are testing it right now.
So guinea pigs, basically.
Yeah, so I'm hoping right around prime cancer age that this sucker's ready to go. All right, DNA oncogenic viruses. We have adenoviridea, herpes viridae, specifically the Epstein-Barr virus, pox viridae, papillomaviridae, and hepadnaviridae, which is hepatitis, papilloma, pox, and herpes. The herpes. Retroviridae, viral RNA is transcribed to DNA via reverse transcriptase, which then can integrate with the cell. HTLV, HTLV1 and 2, can cause adult leukemia and lymphoma. So latent virus remains asymptomatic in hosts via long periods of time, cold sores and shingles. So that's HSV1 and HSV2. A persistent viral infection occurs gradually over a long period of time and is generally fatal. We have this subacute sclerosing panencephalitis. All right, this occurs at a rate of 1 per 1,000 population. So if you have 1,000 people that are not vaccinated for measles, and they all get measles, you'll see 1 death as a result of measles. And that's what this is. It's, brain death, basically. All right, let's talk about prions. Proteinaceous infectious filaments. Everybody, we talked about this in week 1, time to revisit it. Everybody in here has prions as transmembrane proteins in the brain. Occasionally, those prions will just unravel and unfold. When they denature, unravel or unfold, your body will try to remove them. As the body tries to remove them, the body overreacts with an immune response, and it slowly causes brain cell destruction, and your brain shrinks. Mad cow disease is referred to as Creutzfeldt-Jakob's disease variant because the mad cow, the cows, were given, when the ground beef was made, the brain or spinal cord of a diseased cow that they didn't know was a diseased cow was integrated into the ground beef and then sold to you, and then you ate it, and then you died. Creutzfeldt-Jakob's disease, sporadic, just generally occurs after the age of 65, right around 75 on average, randomly, 1 per 1 million people. One day you're going to wake up and you're going to have dementia-like symptoms, and then you're going to be dead 18 months later. There's no treatment, nothing we can do, we just burn the corpse. And burn any medical instrument that was used in your treatment. Gertzmann-Straussler-Streicher syndrome. This is again just another prion disease that originated in Germany. Fatal familial insomnia. This is the genetic version of Creutzfeldt-Jakob's disease. So we have a gene. I don't know if I've gone over this with you guys, but this is kind of dark. So let's pick your dad, because people are less sentimental. So your dad has a gene, this gene, dies at 75. Every time your dad reproduces, so if you have siblings, it's a 50-50 shot that gene is going to be passed on to one of those siblings. But that gene is fundamentally altered. So if he died at 75, the person who received the gene will die at 65. And then if that person reproduces, if the kid gets any of those genes, that kid will die at 55, 45, 35, and it will keep going down until below reproductive age, and then that gene will vanish from the gene pool. It's a very strange disease. They named it because it would wipe out family lines, and they couldn't figure out what was going on. Like, family lines would just disappear. Like, they started dying earlier and earlier until there was nobody left. And then sheep scrapie, which is basically just prion diseases in sheep. If you're a hunter, you know the Midwest is undergoing a problem with wasting in deer and elk, and that is also, wasting is a prion disease within that population. Humans have wasting, that's a different thing, that is not a prion disease. So if you hear wasting in humans and you're like, they're gonna die, no, it's different. Alright, so, this is looking at the normal cellular prion protein on the cell surface and the scrapie protein forming plaques. So the scrapie protein, which is this protein right here, will associate with a normal protein and cause it to denature. And it will keep denaturing. The body will endocytose it, try to dissolve it with hydrogen peroxides, and not be able to until the cell dies as a result of oxidative stress. Viroids are less interesting. Plant viruses enter through wounds via insects. Plant cells generally protected from the disease unless an insect bites it or drinks from it and allows the viroid to appear. This is what they look like, just naked DNA or RNA. And that's it. All right, I'm going to turn this off, and we're going to go over your exam for next week on Thursday. Please be aware, a few things before I pull up the exam. One, the exam will have 50 questions of it, on it. I'm only taking your best 45. Two, you will only have 45 minutes to complete the exam. Then you will have a number on your test. You will break out into groups and retake the test on one Scantron for your group. If your group score is higher than your individual score, apparently. Is it going to stop? I mean, it's going awfully low. Okay. All right. You're going to have a number on your paper. You're going to get together in your group. You're going to retake the test on one Scantron. Okay? If your score is higher from your group than your individual, I will add 10 percentage points to your original score. Full letter grade. If you get a bad group, and your group score is lower, I will still give you a half a letter grade and 5 points. There are no short answers on this exam. You wouldn't have time. Why am I doing it? Those of you who haven't been here before, this exam is the lowest mean average of any exam that I give. It's on average a 52 to a 55%. I have, to solve this problem, eliminated 2 chapters from this unit, moved the test 3 different times, tried to simplify the short answer and make more homework questions from your homework on the exam. None of it has done any good. This is my last Hail Mary pass, hoping that you can all teach each other something that the other one didn't figure out. This is the only test like this. We're going to see if this helps. Okay? All right. What's that? All multiple choice. Yeah. I don't know either. Test isn't that hard.
Can we talk during that group test?
No, it's all interpretive dance.
Really?
Of course you can talk. Alright. Alright, let's go over the test. Yes, obviously, that's the first transcript is. Alright, here we go. In chapter 12, you were provided with a list of vectors and the diseases they cause. Know your vectors and the disease they cause. We talked about, in chapter 6, we talked about bacterial reproduction. We went over a graph and we talked about doubling time. And even in your homework, there were a few problems that focused on the growth curve and doubling time. Spend time in the book, spend time on your homework that had to do with bacterial growth and doubling time. When we're talking about preservation of organisms, there was a few storage methods that could be used. Be familiar with each one of those. Know all of your chemical disinfectants, their mechanism of action, and their preferred use. Know about the different methods of bacterial reproduction. So the conjugation, transformation, and transduction. Know the examples there. When we're talking about regulation of genomes, revisit things like the operator model, the promoter, understand the difference between inducible and repressible. Hopefully that kind of chimes. I think I remember us doing an activity regarding that. Know the process of transcription and translation. What basically is going on. Know how to define archaea, prokarya, and eukarya based on the information that you were presented with in chapter 10. There were 3 specific tables. Understand the biochemical tests and how to interpret them that we went over. Understand how to classify viruses and viral characteristics. Understand the difference between RNA and DNA, RNA positive sense and negative sense. Know oxygen types like tolerances. So aerobic, facultatively anaerobic, obligate anaerobic, microaerobic. We had some fun with parasitic worms. Revisit that section of the lecture specifically. Understand the viral life cycle. So remember attachments, etc., etc. Focus specifically on the animal virus life cycle. Make sure you're familiar with the terms from the malaria life cycle. The terms. Remember, these will be matching and multiple choice, so as long as you can recognize them in context and you know what they mean, you should be fine. know what antigenic shift and drift are. Know what prions are. Oh, no impact of temperature, pH, and osmotic pressure on microbial growth. Temperature, pH, osmotic pressure, those impacts on microbial growth. Now, once again, the exam covers chapters 6, 7, 8, 10, 12, and 13. And I'm not exaggerating when I tell you, out of the 45 questions, at least 10 of them are on chemical disinfectants. So you will not be disappointed with your time spent studying if you familiarize yourselves with those characteristics. All right, I am now entertaining questions, specifics, concepts, scope of the statements.
This is chapter 6, 7, 8, 10, 12, and 13.
Yep, 6, 7, 8, 10, 12, and 13. So if we're looking at, like, from concept perspectives, chapter 6, you're going to spend your time focusing on growth curve and things that interrupt the growth curve and how to store and preserve. Chapter 7, you're going to spend your time specifically with chemical disinfectants. Chapter 8, bacterial genome replication and regulation. Chapter 10, classifying the 3 domains of life and their differences. Chapter 12, hold on, we just went over that. Which was smoking? Oh, life cycles, specifically malaria, parasitic worms, and the vectors that transmit the diseases. Chapter 13, characteristics and classification of viruses, their replication patterns. And prions. Any other questions?
What was it to focus on for chapter 8 that you said?
So so transformation, transduction, and conjugation. So those are the big 3. And then genome regulation, how, like an operon model, and then how DNA replicates. Transcription and then translation. Question?
I just missed my question, so I don't know.
Oh,
It's on Thursday or Tuesday?
should be on Thursday. I looked at the calendar, it should say Thursday. Yeah. But I wanted to make sure I give you even more time with this information, because I don't want you to fail. When I see a student that has like a 90 average for the exams, and I see them get like a 72, there's something wrong. Like, and I've been trying to solve this problem for a very long time, and it just appears to be this test. All right. Are we ready to move on? All right, we don't have a lot of time to do this next bit, so I'm going to yes. Where is the sign-in sheet currently? Thank you. All right. Your paper, which probably should be started soon. General recommendation. epidemiology sections, and your book doesn't give you any guidance on how to write these sections. I am going to do that here. Found out what button I hit.
That color's too light.
What's wrong?
That color's too light. Like all the way back here.
And I'll do this. All right. All right. These two words are where we're going to start this discussion. Prevalence and incidence. Okay, if I said we have 1,000 cases of flu today in Fresno County, does that number mean anything? It's prevalent. Do you know that for sure? Yeah, it is incidence, but we're not there yet. All right. If I said I went through 5 pizzas at the last board game club meetup, does that sound like a lot? What information are you missing? How many people? If I was there by myself and I said I had 5 pizzas, that becomes a big deal. So a rate is what we are talking about here. So a prevalence rate is total number of sick over population. It's a proportion. Incidence is new sick over population. So, let's start with HIV. Each year in Fresno County, we have 100 new cases of HIV. So it would be 100 over the county population size, which is roughly 1 million. So it would be 100 per 1 million population. It gives you an idea. The prevalence of HIV within Fresno County is about 2,000 cases. So that's everybody currently living within Fresno County. So it'd be 2,000 per 1 million. Prevalence only applies if the disease you are talking about has a chronic or long-term state. Influenza is neither chronic nor long-term. So that means the only measurement that you are going to use is incidence. How many people have influenza at this point in time or over the last month? All right, everybody with me so far? So, when we talk about descriptive epi, I need a smaller pen. I'm using up too much. We are looking at incidence or prevalence rates in 3 different categories. Person, place, and time. So, what are person characteristics? Age group. Gender. What else? Occupation. Race, ethnicity. Anybody think any others? Sexual behavior. Let's book into that. Religion. I just misspelled religion. That's fine. All right. Characteristics of place. You guys thinking any? Yeah. Yes. But what are the characteristics of that place that we would be interested in? Urban or rural? What else?
Is it hot or cold?
Climate.
Population?
That's, yeah, population density is urban rural. That's what we're going for. Any other? Developed or developing? This is going to be a weird one. Poverty rate. Okay, so I'm creating a list here. These don't apply to every disease. Every disease has certain characteristics that it's like, oh yeah, this disease has no grouping by age, but it sure as hell has a grouping by occupation. Or like STDs, almost all just age, right? So urban, rural. There are like West Nile virus, for example. You're higher risk for West Nile virus if you're in the country than if you're in the city. Okay? Climate. Certain diseases require certain temperatures. There's a reason why the California flu season is always milder than the rest of the country, because our weather doesn't suck. The colder it is, the more people congregate inside, the higher the transmission rate. So there are different characteristics for each disease. Now this next one, none of you are going to understand, so I'm not even going to ask for questions. Because you're just going to be stumbling around it like a blind person at a nudist colony. Like, all right, here we go. There are four ways to describe a disease trend. We're going to start with the easiest. Season. I already gave you an example of flu. There is a flu season. It has to do with colder weather driving people inside. What you may not be aware of is there is a foodborne illness season. And it starts in May and it ends at the end of August. Summer, people barbecuing. and thinking they know food sanitation and cross-contaminating everything. Your uncle is an idiot. He got the whole family sick. All right. Okay, that's season. Everybody gets the idea of seasons? Okay. Please also understand that certain diseases, childhood diseases, have a season that are directly tied to the first semester coming back to school because the kids have been away. So like pertussis. Pertussis season generally starts in like September and ends in like December. So it's kind of weird and is due to us. Like STDs in Fresno County are 3 days after a winter formal or a prom. They spike to be about, like, and you can see it. So you know when, like, each of the high schools is having an event because STDs follow right after. All right, season. 2, cyclic. Now, cyclic diseases only apply to things that you either get immunity from through recovery from the disease, or you get temporary immunity as a result of recovering from the disease. So measles, before there was a vaccine, was a cyclic disease. That means if somebody brought measles to your community, he would infect everybody in the community with measles. Now everybody is immune for life. 3 to 5 years go by, new people have moved into your community that weren't here when the measles outbreak hit. Babies were born that are not immune to measles. So in 3 to 5 years, the number of susceptibles within your population grows. Somebody, again, comes from the outside, brings measles, and boom, we have another outbreak. Very few diseases exhibit this pattern. 2 of the classic examples are measles and pertussis. We will go over more of why those are the case later, but just know there's very few that meet the cyclic criteria. Third, secular. Secular trends apply to every disease. All a secular trend is looking at the incidence rate of the disease for several years and looking to see if the disease is stable, increasing, or decreasing. That's it. Long-term trends. So they can either be increasing, stable, or decreasing. Number 4. Sporadic. What does sporadic mean? Random. So, you may not know this, but Salmonella typhi, specifically, like the salmonella that killed people in like the 1800s, does not exist in the United States. It's been eradicated. Anytime the U.S. gets a case of Salmonella typhi, it's from travel. So either a traveler came to the U.S. with typhi, or a U.S. resident went out of country and brought typhi back. So you might get 1 case a year, or 3 cases a year. That is sporadic. It does not occur or is transmitted locally, and it's kind of just important. So, in your descriptive epi section, you should have 3 subheadings that say person, place, and time. They should include charts, graphs, bar charts, very little words. You're saying, this is the group most affected. This is the time of year or, whatever the time trend is. This is the place that is most likely. You're saying nothing about risk factors. You're saying nothing about that. You're describing the raw data. Now, what are search terms that you can use to find this data? If you are doing a foodborne illness, you can type in foodborne surveillance reports. If you're doing an STD, STD surveillance reports. If you're doing a respiratory disease, you're going to want to just type in whatever respiratory disease and surveillance. You're getting the idea. It's a surveillance report. And you are looking for characteristics of person, place, and time. It's all statistics, it's all graphs, and it's all math. That's it. I'm not expecting a lot of writing. Everybody clear on this part? Cool. Now let's talk about the part that's going to break your brain. Because everybody loves math. All right. I'm trying to figure out the way to get this to stick. All right, there are about 60-ish people in this class. Let's say that for the rest of your life, half of you have to eat breakfast, lunch, and dinner at McDonald's for free for the rest of your lives. The other half have to eat at Taco Bell for free for the rest of your lives, breakfast, lunch, and dinner. The purpose of this study is we are going to follow you through the course of your lives and count the number of heart attacks in McDonald's versus Taco Bell. So, let's say, remember, there's about 60 of you. So we'll say that over the course of the next 10 years, 5 people from Taco Bell and 5 people from McDonald's had heart attacks. Your ratio of heart attacks is 1. So is there any difference between Taco Bell and McDonald's? No. That is what we call an odds ratio or relative risk. If it's 1, there's no difference. All right, let's keep the math easy. Let's say 10 people that went to Taco Bell had heart attacks, and only 5 people that went to McDonald's. What's your ratio now? 2. That means Taco Bell was twice as likely to give you a heart attack as McDonald's. Now what about this? Five heart attacks at Taco Bell, 10 heart attacks at McDonald's. 0.5. So that means Taco Bell is protective from you getting a heart attack relative to McDonald's. The math doesn't make sense with the example. The example is silly.
The example doesn't make sense, but now it does.
Let me give you an example. Replace Taco Bell with condom use and replace McDonald's with no condom use. So if you use a condom, you are half as likely to get an STD versus not using a condom.
That makes more sense.
Makes more sense? All right. Somebody give me a disease. I need one to work with for this example. Gonorrhea. Well, okay, STDs. Let's go. All right. So if you look at the STD literature, there's a few things that you're going to note as risk factors. One of those things, condom use and availability in the environment. The other is going to be education and awareness. And then they're going to look at socioeconomic factors. And then total number of sexual partners. So as you are looking at those 4 risk factors that we are identifying, they will give you an odds ratio for each one of those to tell you how much more likely each one of those is at predicting whether or not you are going to have or get an STD. We can mathematically figure out your risk. I'll give you another example. There's a disease referred to as, are we out of time? You're out of time. We will pick up this later and then do our activity on Tuesday. You guys are being very polite. Thank you.